We are starting again this webinar on agroecology and gene banks, BRCs. So now I give you the floor, Sandra, and now we will hear about crop dairy systems, some animals at least. Thank you. The floor is yours.
Thank you, Michelle. Welcome to everybody. So my name is Sandra Novak, and I will talk about genetic diversity for agroecology and adaptation to climate change in a mixed crop dairy system. Well, I'm not a specialist of genetics, but I am the scientific in charge of a system experiment named OASIS, which is implemented at a real farm scale on an experimental unit of INRAE in Lusignan, which is a little town near Poitiers in the western part of France. So OASIS is an agro-ecological dairy system adapted to climate change. It's an innovative system breaking from existing systems, and it is experimented at the farm scale. So it's a system experiment. And our main objectives are to permit farmers to live from their dairy system in the context of climate change while saving water and fossil energy resources and contributing to a sustainable agriculture. So to achieve all these objectives, we used an agro-ecological approach, which allows us to valorize the natural resources and also all the dimensions of space and time. And the idea was to design a consistent mixed crop dairy system by strengthening the relationships between the plant and animal components of the system. So we first build a system by diversifying forage resources. That means diversification of plant species, of cultivars, and also mixtures of plant species and cultivars. Also multi-year cropping and, for instance, agroforestry. We also have long crop rotations, and we use drought-adapted crops, for instance, sorghum, and also we use a lot of legumes instead of mineral nitrogen fertilizers. And then we devised a productive and robust herd with a management strategy consistent with this diversified forage system. And here the ideas are to give priority to grazing, to implement 2 calving periods, and to extend the lactation length and also the co-lifetime performance. And so we use 3-way rotational crossbreeding with Holstein, Scandinavian Red, and Jersey breeds. And the relationships between the plant and animal components are reinforced through the recycling of effluents and also the use of dual-purpose crops that can either be grazed or harvested. And the main innovations of this new dairy system are first the diversification of its plant and animal components, and also diversification of some functions of the system with dual-purpose crops or trees. And secondly, the valorization of new dimensions in space or time. For instance, the use of trees to valorize the vertical space dimension, and the use of crop rotations without bear sow or milk production considered at the co-lifetime instead of per lactation only. And the hypothesis we test with this new dairy system is that the increase of diversity in a dairy production system allows to consider good production levels and high environmental performance and to improve the resilience of the whole system. So this system is experimented since 2013 through a long-term system experiment in Lusignan at an INRAE facility with 90 hectares of temporary grasslands and annual crops, and with 72 dairy cows. And in this region, there is a lack of grazed forage in summer because of drought. And it's a low input system with no irrigation and a very limited use of concentrates for animal feeding and of mineral nitrogen fertilizers for plants, and also not a lot of use of pesticides. The aim of this forage system is to achieve forage self-sufficiency for the dairy herd of 72 dairy cows and replacement heifers. So to assess if the objectives of the system are fulfilled, we measure and observe a lot of parameters regarding the weather and agricultural practices, also agronomic and zootechnical performances, for instance, crop yields and quality, grazing practices, daily feeding amount and individual milk production quality, cattle conformation, reproduction and health, and also data available regarding environment, on water and energy consumptions, on biodiversity, on soil fertility, and also economic data, for instance, costs, incomes, and subsidies. And now I will detail more where genetic diversity is used in our agro-ecological mixed crop dairy system. There are 4 components that involve genetic diversity in oasis: animals, plants, trees, and soils. First, I will talk about the genetic diversity in the dairy herd. We have implemented a 3-breed rotational crossing to valorize heterosis. And these are dairy breeds that were chosen for answering to the objectives of our system. So we chose Holstein for its ability to produce a lot of milk and to adapt its production to different kinds of forage. The Scandinavian Red was chosen for its good production and reproduction performances. And the Jersey breed was chosen because this breed has a good reproduction. It is robust regarding diseases and climate conditions. And with its small size, it can also graze when the bearing capacity of soil is not so good. And we choose also it because this breed produces milk with a high amount of solid content, which is interesting for economic and also for environmental aspects. This drawing gives you an outline of the way crossbreeding is implemented in Oasis. We began in June 2013 with our initial Holstein herd, and we decided to test 2 ways of crossbreeding. One by beginning with Scandinavian bred bull semen, and the other one by beginning with Jersey bull semen. So we use artificial insemination for the reproduction of the cows. And as you see, it takes many time to produce a 3-way crossed herd. And our goal is to produce a dairy herd more adapted to the grazing of diversified resources and to climate change, less sensitive to diseases, and with a good reproduction to limit unproductive animals. And here are some results on milk production, but only for the F1 because we have not so much lactation for the other breeds, crossbreeding. As you see, Holstein crossed with Jersey in blue produce more fat and protein contents in their milk than Holstein crossed with Scandinavian bred. So what is the link with the BRC? Here, the BRC concerned is the bridge platform from INRAE. And we sent to this platform blood samples of all our cross dairy cows. And our goal is to have a long-term storage of their DNA, which is available to scientists who could like to study this information in relationship with the other data that are measured on the herd or on the farm scale. And now let's talk about genetic diversity that is present in oasis on crops and grasslands. So on this slide, you can see the plots and the 3 crop rotations that are used for the cropping system of oasis. So I will not go into the details, But as I explained at the beginning, there is a lot of diversity in crops and grasslands. All grasslands have different compositions, and there are multi-species grasslands. And on 4 plots, we have also alley cropping agroforestry. And as I said before, our objective is to produce the forage necessary to feed our dairy herd and their replacement heifers. And here are 2 examples of genetic diversity we have implemented. So all of crops are a mixture of cultivars. For instance, we used this year 5 cultivars of sorghum bicolor, differing by their height and earliness. And on the right side, you see a multi-species grassland, where we have sown 2 cultivars of tall fescue. And we use genetic diversity to be less sensitive to hazards or diseases. And currently, we have no link with BRC because as we work at the field scale, we need to use a large amount of seeds, but also because commercial seeds have interesting performances. And in our cropping system, we do not design new cultivars. But, well, in fact, we do not study it. But maybe our low input system could generate genetic diversity in grass species, but we do not study that yet. Well, what about the genetic diversity of trees? On Oasis, we have mainly planted fodder trees, but we have also timber trees. So on this plot, we have pollarded the trees so that they can be browsed by the dairy cows. That means we cut the trunk at 50 or 80 centimeters. And this is a white mulberry. We have also 4 other species. And white mulberry, we use the seedlings from a commercial tree nursery. And we observed a great variability in the shape of leaves and production levels. But we do not know, or we do study their genetic diversity. But we think that this could influence their palatability or even their nutritive value. So the genetic diversity here has not been chosen. And well, currently we have no link with BRC because no BRC exists on this thematic. And this is another plot where we have planted 3 species of high stem trees to provide timber. And these are wild white cherry, service tree, and wild service tree. And we test 1, 2, or 3 seedlings per planting location. And for wild cherry, we have planted 2 progenies and 1 cultivar. And this study is led by Frédéric Santi, who will do the next presentation. So I will later explain the link, potential link with BRC. And finally, let's talk about genetic diversity that is studied in the soils of Oasis. So in order to study the evolution of the soil characteristics that are induced by the implementation of our low input system, and also in order to compare these evolutions on cropping systems that are either grazed or harvested, we collected soil samples in 2016 and 2021 this year on 6 plots. And several soil characteristics are studied, in particular soil microbiology. And for this thematic, we work with the platform Genosol of INRAE in order to store on the long term the microbial genetic resources of our soils. And these resources are also available to scientists. So in conclusion, genetic diversity is used in OASIS to enhance resilience to hazards, that can be diseases, pests, or climate events, in the context of a low input farming system with less pesticides, less concentrates, nitrogen fertilizers, and veterinary products. And as we have seen, our long-term agro-ecological system potentially generates genetic diversity in soils and maybe all and in grass species. Our actual links with BRCs are mainly relative to animal and soil microorganisms genetics in order to store on the long term the biological resources that are generated by our system. And these resources are available for scientists to better study the processes linking genetic variability, agroecology, and adaptation to climate change. So many thanks for your attention, and if you have questions, I'm ready.
Thank you very much, Sandra. So in terms of questions, yes, we have a question in the room. So
thank you for the very interesting presentation. You were speaking about resource available for scientists. Do you also communicate the information to farmers in general, or make the farmers more sensitive and aware of the necessity to have a better approach, if I may say?
Yes, thanks for your question. Yes, we have a lot of visits from farmers and also from advisors, and there is no a network of farmers and advisors interested in climate change, because it's an important issue for our farmers, and they are interested to know new solutions to have a forage self-sufficiency, because it's very difficult in our region these last years, the much of droughts and so.
Alors, there is a question from Gwendal, so you can reach the floor.
Yeah. Thank you, Sandra, for this very nice presentation. You decided to use crossbreeding for your dairy cattle, and I'm just wondering if using instead multiple breeds can enhance the use of the complementarity of the crops. You know what I mean? Having different ecological niche or different preferences toward one or other crops or trees, given the fact that you have a lot of variability in trees or in the crops.
I'm not sure I really understand your question.
I mean, here you use only one breed, in fact, which is a crossbreed, in fact, between 3 different breeds of dairy cattle. And thus you've got a large variability of individuals within the same breed. But I'm wondering if you are using 3 different breeds instead, with 3 different flocks, they can be attracted by different part of the crop of your system, of your agronomic system.
Well, we use crossbreeding because of we were also interested in the heterosis effect.
Yeah.
And we don't think that, for instance, Holstein is very adapted to our system. And we really wanted to have all the abilities, for instance, good reproduction, also good production, also not to be sensitive to head stress. So we want all these capacities, so that we have done crossbreeding.
Okay, because my question was about the impact of complementarity of your crops and the better way to use this complementarity, having more or less different dairy system, in fact, because they are more specialized on, I don't know, grazing herbs for one of the breed, and another breed more attracted by trees, or these kind of things. You know what I mean? It's
yes. Well, I'm not sure.
hey, I don't have any answer. It's just a question.
A giraffe, but maybe it could be to you another species, not another breed. But
okay, no,
that would be more, yes, more interesting to use another animal species as goat or not giraffe. But
if you want. I'm not sure it's fully adapted, but
to produce milk, I don't know, but
yeah, why not? Okay, thank you.
Welcome.
So there is another question from Monica, and then you can go on stage also.
On the chat. Yes, I just read the question. Results from your one generation.
Okay, it looks like I'm able to connect to you. That's good. Yes, there was a little bit shown at the beginning with the F1 generation and the milk production and solid content. I just wondered, so you have like theoretically basically chosen 3 different breeds that together would somehow give you the traits that you want for your location. And I wonder whether you've been successful in achieving that, or whether you don't know yet. Maybe it's too early.
It's a little bit too early, but what the technicians observe is that we improve all the characteristics that we were interested in. That means reproduction and less health diseases. But in fact, what is also important in Oasis is that we have designed a consistent system, and we have mixed all the different strategies that are also extended lactation and 2 calving periods. So all the effects are mixed, and we have also less concentrates. So everything I think will contribute to these improved performances, and we do not really know which individual factor improved this performance.
Sorry,
or other factors.
Yeah. Just showing my ignorance, can you answer 2 very small questions for me? What's meant by concentrates, and how many calving periods are normal? I'm not a cow specialist.
Concentrates are, for instance, Brazilian soya, which is used to give some nitrogen to the ratio to the feeding of the cows, to equilibrate, to balance it with the energy. And normally the farmers use a lot of concentrates. And about the other question, normally there is only 1 calving period, or there is no calving period. And we have put our calving periods on the periods where the grass is more growing. So in spring and autumn. So because we want to increase the grazing period of in the feeding of our cows.
Okay, thank you.
You're welcome.
Thank you very much. I have one question about the feedback you get from the farmers when you said you have visits.
Yes.
And regarding the performance of the flock, either the milk quantity or economic profit, because when you showed the performance for milk, it was comparing the 2 types of crossbred, but how does it compare with the average result of the farmer in the surroundings, in the same area of your flock?
Yes, we have assessment at the level of the farm. And we also did assessment on the economic parts of the, and so we showed that our system is interested also at the economical level. And what was interesting is that the solid matter produced is the same than at the beginning of the experiment. And the milk production has decreased, but the solid matter remains constant. And we use less concentrates, like less inputs. So it's interesting economically. And that's the farmers are very interested in these results.
Yeah, sure. Okay, thank you very much, Sandra.